Forensic examination of hair. Hair is encountered as physical evidence in a wide variety of crimes. It is not possible to individualize a human hair through its morphology or structural characteristics, but through the isolation and characterization of DNA present in the hair. However, the morphology or structural characteristics of hair can provide strong corroborative evidence for placing an individual at the crime site. Hair is an appendage of skin that grows out of the hair follicle. The length of hair extends from its root or bulb embedded in the follicle, continues into the shaft, and terminates at the tip end. The shaft is composed of 3 layers: the cuticle, cortex, and medulla. The cuticle enables hair to resist chemical decomposition and retain its structural features over a long period of time. The cuticle is a scale structure covering the exterior of the hair. The overlapping scales are formed from specialized cells that have keratinized and flattened, and they always point to the tip end. The scale pattern is an important feature for species identification. The cortex is the main body of the hair shaft and is contained within the cuticle. The cortex is made up of spindle-shaped cortical cells and is embedded with pigment granules that give hair its color. The color, shape, and distribution of these granules provide important points for comparison. The medulla is the central canal running through a hair and is made up of a collection of cells. The presence and appearance of the medulla vary from individual to individual and even among the hairs of a given individual. The medullary index measures the diameter of the medulla relative to the diameter of the hair shaft and is normally expressed as a fraction. For humans, the index is generally less than 1/3. For most other animals, the index is 1/2 or greater. The medulla may be classified as being either continuous, interrupted, fragmented, or absent. In human head hairs, the medulla is usually absent or fragmented. Most animals have a medulla that are either continuous or interrupted. In addition, the shape of the medulla in animals vary from nearly cylindrical appearance to a patterned shape. The root and other surrounding cells within the hair follicle is important for hair production and growth. When pulled from the root, some hairs have a follicular tag, which is the richest source of DNA associated with hair. Forensic hair examiners can individualize human hair by characterizing the nuclear DNA in the hair root or follicular tissue adhering to the root. Human head hair grows in 3 developmental stages: anagen, catagen, and telogen phase. In the anagen phase, or the initial growth phase, the root is attached to the follicle for continued growth. When pulled from the root, some hairs in the anagen phase have a follicular tag. Hair continues to grow, but at a decreasing rate during the catagen phase. Once hair growth ends, the telogen phase begins and the hair is pushed out of the follicle, causing the hair to naturally shed. Most often, the prime purpose for examining hair evidence in a crime laboratory is to establish whether the hair is human or animal in origin. A careful microscopic examination of hair reveals morphological features that can distinguish human hair from animal hair. Scale structure, medullary index, and medullary shape are particularly important in hair identification. In comparing human head hair, the criminalist is particularly interested in matching the color, length, and diameter. Other important features are the presence or absence of a medulla and the distribution, shape, and color intensity of the pigment granules in the cortex. A microscopic examination can also distinguish dyed or bleached hair from natural hair. A dyed color is often present in the cuticle and cortex, while bleaching removes pigment from the hair and gives it a yellowish tint. When a questioned hair does not have adhering tissue or root structure, the criminalist can analyze mitochondrial DNA, which is found in larger quantities than nuclear DNA in our cells, including cells in the hair shaft. Unlike nuclear DNA, mitochondrial DNA is only transmitted from mother to child. Therefore, it cannot distinguish hairs from different individuals who are maternally related. Hairs from different parts of the body vary significantly in its physical characteristics. The questioned and standard or reference hairs must come from the same area of the body. It is also important that the collection of the standard or reference sample ensures a representative sampling of the hair from any one area of the body. Collecting 50 full-length hairs from all areas of the scalp normally ensures a representative sampling of head hair. A minimum collection of 24 full-length pubic hairs covered a range of characteristics present in this type of hair. During an autopsy, hair samples are routinely collected from a victim of suspicious death.